Hello guys, welcome to our YouTube channel. So today we are going to continue with the lectures on blood physiology. My name is Fabius Masibo. Remember to subscribe, like, comment and share with your friends. So I hope you enjoy. I will go ahead. Last time we were talking about the introduction to blood physiology. So today we are continuing to look at the components of blood. So today we are going to talk about the plasma proteins, then we'll go ahead and talk about the red blood cells, then look at the process of erythropoiesis, and so on. So welcome guys, I will start. So when you talk about the plasma proteins, uh, what comes in your mind is that you just remember the three major plasma proteins that include serum albumin, serum globulin, and fibrinogen. Yeah, as you can see, like we have serum albumin, serum globulin, and fibrinogen. Fibrinogen is not contained in serum, that's why we don't name it as serum fibrinogen. It's just fibrinogen. Yeah. So normal values of these plasma proteins, like for the case of albumin, the normal values are 4.7 grams per deciliter. Serum globulin is 2.3 grams per deciliter. Fibrinogen, it is 0.3 grams per deciliter. So what are the major properties of these plasma proteins? So the specific gravity is around 1.026. Then these plasma proteins, they are responsible for the oncotic pressure in blood. Yeah, the normal oncotic pressure in blood, it's usually 25 millimeter mercury, and albumin plays a major role in the maintenance of this oncotic pressure. Yeah, out of the three, albumin contributes a lot. Then the plasma proteins also contribute to 1/6 or a sixth of the total buffering action of blood. Also, in this case, albumin plays a major role. Yeah, then among the three plasma proteins, the fibrinogen is the one that has the highest molecular weight. Yeah. Then where do these plasma proteins come from? Like where are they synthesized or where are they produced? So in the embryo or in the fetus, they are synthesized by the mesenchymal cells. Yeah. Then in adults, they are synthesized mainly from the reticuloendothelial cells of the liver. Yeah. Then you should know that these two, like when you're talking about the plasma proteins, like we said, we have globulin, we have albumin, and fibrinogen. So globulin, they are not synthesized by these two, like they are not synthesized by the reticuloendothelial cells. So globulin is different because they are synthesized from the B lymphocytes. Yeah. So what do they do? What do the plasma proteins do in your body? So the first thing is that they have a role in the defense mechanism of the body. In this case, we are talking about the gamma globulins, which act as antibodies in the defense mechanism to protect yourself or your body from invading agents. Then they have a role in blood coagulation. In this case, we are talking about the fibrinogen, which is a clotting factor. Then they have a role in transport mechanism of our body, in the body. For example, albumin, it's able to transport substances like drugs, among other things. Then we said they maintain the osmotic pressure in blood. Majorly is albumin. Then they have a buffering mechanism of blood. Yeah. Then albumin is the major plasma protein here involved in the buffering mechanism. Then they have a role in the provision of viscosity of blood. Yeah. Then in this case also albumin plays the major role. Yeah. So the plasma proteins vary. Yeah, they can be increased or decreased. So when they're increased, they are called hyperproteinuria, or when they're decreased, when they're below the normal ranges, yeah, they are called hypoproteinuria. Like protein means proteins, the proteins, yeah. So the cases whereby you'll find hyperproteinuria is like when a patient is dehydrated, uh, hemolysis, like destruction of either red blood cells or the blood cells, the plasma proteins are decreased. Acute infections like hepatitis and acute nephritis, you'll find that the plasma proteins levels are elevated. In rheumatoid arthritis, yeah, in alcoholism, in excess of glucocorticoids, yeah, in leukemia, in respiratory distress syndrome, yeah, among others. Then you'll find hypoproteinuria, like reduced levels of plasma proteins in patients, maybe, or in people who are having diarrhea, hemorrhage, yeah, who have had burns, who are pregnant, yeah, who are malnourished, yeah, or those who have prolonged starvation or starved, in people who have cirrhosis of the liver or liver cirrhosis, yeah, in chronic infections like hepatitis also, yeah. So that's all about the plasma proteins. So I'm going to talk about the cellular components of blood now. The cells, we said blood is a connective tissue. So, and you know, a connective tissue is made up of the cells and the extracellular fluid. So the fluid we have been talking about is the plasma, and the components of plasma we have said. So now we want to go to the cellular components of blood, whereby we'll start with the red blood cells, RBCs. Yeah, they're also called erythrocytes. Erythro means red, like that's why they're called red blood cells, because they are red. Then we look at some of the properties of these red blood cells. They are uh, normal blood count, morphology, yeah, and their functions in blood. Then we look at the process of formation. So red blood cells, these are non-nucleated, so that they don't have the nuclear. Yeah. Then they are large in numbers. Yeah, we'll see that in millions. Yeah, per cubic millimeter of blood. So compared to other blood cells like white blood cells and platelets, this one they are large in numbers. Then they are red because they contain a coloring pigment called hemoglobin. They are responsible for transport of respiratory gases like carbon dioxide through the lungs, yeah, using hemoglobin, and oxygen to the tissue also using hemoglobin. We'll look at the mechanism. So what is the normal red blood cell count? So the normal red blood cell count is 4 million to 5.5 million per cubic millimeter of blood. This number varies according to the sex, like male, they tend to have around 4.5 to 5.5 million, an average of 5 million per cubic millimeter of blood. Then females, they tend to have a slightly lower, like around 4.5 million cubic per millimeter of blood on average. So what is the normal morphology of the red blood cell? They are disc shaped and biconcave. We'll see the advantages. So the advantages of but the biconcave shape of the red blood cell, one is that the biconcave shape helps in equal and rapid diffusion of oxygen and other substances into the interior of the red blood cell. Another advantage is that the biconcave shape provides a large surface area, yeah, for absorption or removal of different substances. Then number three is that the biconcave shape helps to provide minimal tension, yeah, on the membrane when the volume of the cell is altered. Yeah, then another advantage is that because of this biconcave shape, uh, it offers like advantage, especially when passing through small capillaries. Yeah, you know, the red blood cell will squeeze through, will be changing the shape, yeah, very easily without getting damaged. Yeah. So what is the normal lifespan of the red blood cell? Average lifespan is about 120 days. After this lifetime, like the old or also called senile red blood cells, that are destroyed in the reticuloendothelial system, that's in the spleen, majorly in the spleen. So the properties of red blood cells, yeah, they have a specific gravity of 1.092 to 1.101. They have a packed cell volume. So packed cell volume is the proportion of blood that is occupied by the red blood cells and is expressed in percentage. So the normal packed cell volume, it's 45% of blood, and the plasma volume is 55. When you add this, you get 100, because we said you express it in percentage. Then red blood cells, they tend to form a Rolex. Yeah. So this Rolex formation is a property. Like you'll see that the red blood cells are piled up together like coins. So this is facilitated by the plasma protein, fibrinogen, and globulin. Yeah, I hope you remember the plasma proteins. Yeah. So what are the major functions of the red blood cells? What do they do in the body? The major function you said is the transport of respiratory gases. So red blood cells, they transport oxygen from the lungs to the tissues. Two, they transport carbon dioxide from the tissues to the lungs. We'll see that oxygen combines with hemoglobin to form hemoglobin. Then it takes that oxygen from the lungs to the tissues whereby it's exchanged there. Then the same thing to carbon dioxide will bind to hemoglobin, yeah, then it will be taken to the lungs for exchange, yeah, to be expired out. Also, the red blood cells, they have a role in the buffering action in blood, yeah. Then in blood group determination, whereby here we say like the red blood cells, they contain the antigens, antigen A and antigen B, yeah. Using those antigens, we are able to group the blood, like we are able to group the blood types or blood groups as either blood group A, blood group AB, or blood group O, yeah, depending on those antigens. Yeah, so this is the fate of the red blood cells. So we say old or semi red blood cells, they are destroyed by the reticuloendothelial system, of which majorly is the spleen. So after their lifespan has ended, they are destroyed in the spleen. So uh, the red blood cells are made up of hemoglobin. Yeah, we say they are nucleated, but the major component they are made up of is hemoglobin. So after they are destroyed, they will release hemoglobin. Yeah. So this hemoglobin is made up of iron and globin and porphyrin. So these three make up the hemoglobin. So after hemoglobin is broken down, it will release globin, iron, and porphyrin, I mean. Yeah. So iron will be taken by apoferritin, yeah, or will combine with apoferritin to form ferritin, and then it is stored to be reused in the synthesis of hemoglobin again. Then globin, that is broken down like it's directly goes to the protein pool and stored, whereby it will be also be reused. Then porphyrin, yeah, this one it will be converted into bilirubin and excreted. Yeah, we'll look at that when we come to the synthesis of hemoglobin. Yeah. So what is the process of erythropoiesis? Like how are the red blood cells synthesized? So erythropoiesis is the process of origin, development, and maturation of the red blood cells. Yeah. Uh, we can be able to define another process called hematopoiesis, yeah, to differentiate it from erythropoiesis. So hematopoiesis is the process of origin, development, and maturation of all the blood cells. Erythropoiesis, remember we said erythro is red, so it's the process of synthesis of red blood cells. But hematopoiesis, we are talking about all the blood cells. So the site of erythropoiesis, yeah, so we can divide it into two, in fetal life and in adult or newborn or children's others. So in fetal life, the red blood cells are produced in the mesenchyme of the yolk sac during the first 2 months. Then from the third month, the liver becomes the major organ for production of the red blood cell. Then during the last 3 months, like from the 6th month to the 9th month or to delivery, the red bone marrow and the liver are the major organs that will be producing the red blood cells. Yeah. Then in newborn babies, growing children, and adults, the red blood cells are produced only from the red bone marrow. Yeah, I hope you understand that. So this is the process of erythropoiesis. So first of all, we start with the stem cells. Yeah, stem cells, like stem cell, it means it can be able to produce another cell. Yeah, it can be able to divide to produce another cell, or it's the stem of production of the cells. Then hematopoietic, it means blood, like hemo is blood, so it's the cell that produces the blood cells. Then pluripotent, it means it can produce many cells. Then we start with the uncommitted, yeah, uncommitted pluripotent hemopoietic stem cell. It means it's not committed to producing one type of blood cell. Like it can produce any of the blood cells. It can produce either the red cells, white cells, or platelets. From that step, like it must be committed now. Like it must be committed. It must be decided to either produce one of the or a specific type of blood cell. So in this case, it can be committed to produce either the lymphoid, like, so the one that will be committed to produce the lymphocyte, it will be called lymphoid stem cell. The one that will be committed to produce the other one, like the other three, will be called colony-forming blastocyte. It's committed now to forming all the colonies. Yeah. So colony-forming blastocyst, uh, it divides into colony-forming unit erythrocyte. So colony-forming unit erythrocyte is the one that will be producing the red blood cell. Next time we'll be looking at this other colony-forming unit, for example, granulocyte, mega granulocyte, monocyte or monoblast, I guess, or colony-forming unit megakaryocyte, which will be producing the platelets. So our main focus today is colony forming unit erythrocyte, of which it will be producing the red blood cell. So this is a better explained diagram. So we started by saying we have the pluripotent hemopoietic stem cell, which will differentiate into colony forming blastocyte. So colony forming blastocyte, we are interested in colony forming unit erythrocyte, the one that will be producing the red blood cell. So it will start and differentiate into proerythrocyte. Proerythrocyte, you can see the shape, it's big, yeah, it contains the nucleus, yeah. Then proerythrocyte will differentiate into early normoblast. You can see the shape or the size has decreased. Yeah. Then intermediate normoblast, you can see the changes. It has started being red because of hemoglobin. Yeah. Then the late normoblast, you can see the nucleus is not there anymore. Yeah. Then the reticulocyte, then the red cell. So let's look at the major events that occur in these phases. Yeah. So the major event that occurred during pro-erythroblast phase of is the synthesis of hemoglobin starts. Yeah, you remember here the synthesis of hemoglobin starts. Then early normoblasts, the nucleoli disappear. Then in intermediate normoblast, the hemoglobin starts appearing now. You can see the changes, like the cell starts being red. Then in late normoblast, the nucleus disappears. Yeah, then in the reticulocyte, the reticulum is formed. Yeah, then the cell enters capillary from the site of production. Yeah, so the major event that occurs here is whether, like, if it was being formed in the red bone marrow, it moves and enters the circulation now. Yeah, then the matured red blood cell, yeah, the reticulum disappears, then the cell become biconcave. I hope you have understood. Yeah, we are done. Thank you so much. Thank you so much. Remember to subscribe, like, comment, and share. So next time we're going to look at the white blood cells. Yeah, we're going to look at the white blood cells, and as we go on. Thank you so